So, let's go. Number 1. Lola's chewing gum. On the muddy shore of a Baltic lagoon, on the island we now call Lolland, a young woman worked a black lump of birch tar between her teeth until it turned soft, then spat it into the mud and walked away. That was 5,700 years ago. She never knew that the wad she discarded would outlast her bones, her people, and her entire world, and that it would one day give a laboratory in Copenhagen her whole body, from the color of her eyes to the germs living in her mouth. The site is called Seal-Borne, uncovered during construction of the Great Tunnel Link across the Fehmarn Belt. The material she chewed was birch pitch, a sticky black substance made by heating birch bark until it weeps a tar. Across prehistoric Europe, people chewed this pitch to make it pliable, using it as glue to fix stone blades into wooden handles. It may have soothed toothache, and its natural antiseptic compounds may have cleaned the teeth. And because pitch seals out air and water, and because it's soaked in a young woman's saliva before sinking into oxygen-poor coastal mud, it preserved something almost no burial ever does. In 2019, a team led by Theis Jensen and Hannes Schroeder at the University of Copenhagen published their results in the journal Nature Communications. From that single chewed lump, they recovered a complete ancient human genome. It was the first time a whole human genome had ever been pulled from something other than bone or teeth. The chewer was a woman, and researchers nicknamed her Lola. Her genes told them she had dark skin, dark hair, and striking blue eyes, the same combination carried by other Western hunter-gatherers of the time. She was more closely related to those foragers than to the farmers, then spreading across Europe, a hint that older ways of living had not yet vanished from the north. Then the analysis went further. The pitch had trapped Lola's oral microbiome, the community of bacteria and viruses living in her mouth. Among them was Streptococcus pneumonia, a leading cause of pneumonia and meningitis, still one of the deadliest bacteria alive today. There was Porphyromonas gingivalis, which rots the gums. There was even the DNA of a herpes virus. Alongside her germs lay traces of a recent meal, hazelnut and mallard duck. In one discarded object was a woman's ancestry, her last supper, and the diseases she carried, all at once. Consider how improbable that survival was. DNA begins to break apart the moment an organism dies, sliced into ever shorter fragments by water, heat, and microbes, which is why most ancient genetic material is a ruin. Birch pitch works against all three enemies. Its tarry compounds are mildly antiseptic, suppressing the bacteria that would otherwise consume the genetic traces. It repels water, shedding the moisture that drives decay. And once it sank into the cold, oxygen-starved mud of a submerged coastal lagoon, it was effectively sealed in a time capsule. Seal-Borne itself was a shallow, brackish inlet, the kind of waterlogged environment that preserves organic material for thousands of years. The researchers noted that Lola's genetic profile placed her among people who still lived by hunting, fishing, and gathering, at the very moment farming was arriving in southern Scandinavia, a reminder that the old world and the new overlapped for centuries rather than switching over in a single generation. Here's why that matters, far beyond one Neolithic girl. The pneumonia bacterium found in Lola's 5,000-year-old spit is not extinct. It is the same lineage circulating through daycares and hospital wards right now. Her chewing gum is a message from the first pages of the human disease story, before anyone could write a single word of it. And to read those pages, we have to look where no one thought to look, which brings us to a tomb full of people who all died at once. Number 2. The Roaix Epidemic Tomb. A shaft of pale light falls through a narrow opening into a rock-cut chamber in the hills of Provence, and it lands on a floor carpeted with human bodies, packed shoulder to shoulder, laid down not one by one over years, but all together, in what looks like a single terrible day. This is the Hippodrome of Roaix, and archaeologists have argued about that floor for more than half a century. Roaix sits in the Vaucluse, in southern France, and it belongs to the Chalcolithic, the Copper Age, dating to somewhere between 2500 and 2000 years before the Common Era. It was excavated in the 1960s by the French prehistorian Jean Courtin, and it became the type site for a whole regional culture, the Roaix group. What made it famous was its structure. The tomb held two separate burial deposits, divided by a clean layer of sterile sediment. The lower deposit looked ordinary, the successive burials of a community, adding its dead over many years, the normal use of such collective tombs. The upper deposit was different. It was a dense mass of skeletons, men, women, and children together, with little of the layering that slow, repeated burial produces. Estimates run to 100 individuals, or more, interred in what appears to be one event. Courtin called it the niveau de guerre, the war level, because some of the bodies had flint arrowheads lodged in their bones. Here is where the tomb turns from a curiosity into a genuine mystery, because those arrowheads point one way, and the sheer number of dead points another. If people were killed by arrows, this was violence, a massacre, or a battle that wiped out a community in an afternoon. But a mass grave filled all at once is also exactly what an epidemic leaves behind, when the living cannot bury the dead fast enough and simply lay them together. The Copper Age in this region was a tense time, with fortified settlements and clear signs of rising conflict, so violence is plausible. Yet disease cannot be ruled out, and some of the dead may have been buried in panic during an outbreak, with a few victims of a raid mixed in. The arrowheads themselves were pressure-flaked flint, painstakingly shaped and hafted onto wooden shafts, the standard weaponry of the Copper Age in this corner of Europe. This was also the era when bell beaker material culture was spreading across the western half of the continent, a time of movement, new metals, and new tensions between groups. Collective tombs like Roaix were used and reused across generations, which is precisely what makes the upper level so hard to interpret, because a chamber that normally filled slowly, body by body, was instead filled almost at once. That contrast between the patient lower deposit and the sudden upper one is the heart of the puzzle. Whatever struck this community did not give it time for its usual rites. The honest answer is that without ancient pathogen DNA, which the old excavation could not recover, we cannot say for certain. Mainstream readings favor warfare. Others see a mixed catastrophe. A few suspect an epidemic burial dressed up as a battlefield. What is not in doubt is the horror the floor records. A whole community, every age and both sexes, put into the ground in a single act. That is the moment a village ends. And the thing that most often ends a village without leaving a mark on the bones is something you cannot see at all. Number 3. Parvovirus B19. In a Bronze Age grave, a skeleton lies quiet in the soil, and inside the dense core of its bones, in the tissue that once made blood, a virus still waits, its DNA intact after thousands of years, ready to tell scientists about an outbreak nobody knew had happened. The virus is Parvovirus B19, one of the smallest viruses that infects us. It is a single-stranded DNA virus, and it has a very specific target. It attacks the young cells in the bone marrow that mature into red blood cells. In most children, it causes a mild illness called fifth disease, known for the bright red slapped cheek rash. In adults, it can bring aching joints. But in certain people, it is genuinely dangerous. In a pregnant woman, it can cross to the fetus, destroy its ability to make blood, and cause severe anemia, fluid buildup, and miscarriage. In people with sickle cell disease or weakened immune systems, it can shut down red cell production entirely, a crisis that can kill. Because B19 is a DNA virus, and DNA survives far better than the fragile RNA of many other viruses, its genome can persist in ancient bone. In a study reported around 2023 and 2024, researchers including teams at the Francis Crick Institute reconstructed dozens of ancient B19 genomes from human remains across Eurasia. The virus turned out to have infected people for more than 10,000 years, since the early Holocene. But the striking finding was not just its age. It was a dramatic change during the Bronze Age. Around that time, the diversity and spread of the virus appear to surge. And that surge lines up with one of the great upheavals of European prehistory, the mass migrations out of the steppe, the movements associated with the Yamnia and their descendants, when whole populations poured westward and mixed with the people already there. A virus spread by coughing, by blood, and from mother to child would have thrived on that churn of bodies. New populations met new pathogens, and old viral lineages died out as new ones took over. The ability to reconstruct these genomes at all depends on where the virus hides and how it is preserved. B19 concentrates in the bone marrow, and the densest bones in the body, the petrous portion of the skull around the inner ear, and the roots of the teeth, are the richest reservoirs of ancient DNA. From tiny quantities of surviving fragments, researchers can piece together whole viral genomes and then run a molecular clock backward, estimating how fast the virus mutates and therefore how long its lineages have existed. The picture that emerges is of a virus with real staying power, persisting through population turnovers that erased other lineages entirely. The work runs in parallel with ancient studies of hepatitis B, another durable DNA virus recovered from prehistoric remains, and together they show how a class of quiet, chronic infections has shadowed humanity for far longer than any written record admits. Think about what this means. A virus we now file under mild childhood illness was riding the great migrations of the Bronze Age, and everywhere it went, it was quietly killing the unborn and draining the blood of the vulnerable. For 10,000 years, it left no record anyone could read until we learned to read it in the marrow of the dead. And migration was not the only force breeding new killers. Something even more basic to human life was doing the same thing down in the villages where people had only just learned to farm. Number 4. The first farmer's fever. In a cluster of mud brick houses, beside a pen crowded with the first domesticated animals, a person lies shivering with a fever that has climbed into the bloodstream. A systemic infection that a few thousand years from now, doctors will call enteric fever, and the bacterium burning through them has only just learned how to make a human this sick. That bacterium is Salmonella enterica, and the particular form is the Cerevar paratyphus C, which causes paratyphoid fever. This is not the brief food poisoning most people picture when they hear salmonella. This is a systemic infection that invades the bloodstream and can be fatal without treatment. And in 2020, a study in the journal Nature Ecology and Evolution, led by Felix Key at the Max Planck Institute for the Science of Human History, traced its origins deep into prehistory using ancient DNA. The team recovered salmonella genomes from human skeletons spanning roughly 6,500 years across Eurasia. The oldest came from Neolithic and Bronze Age people, including early farmers and steppe pastoralists, and the genomes recorded a transformation. The bacterium's genes showed progressive decay, whole sections lost or broken, which is the classic signature of a microbe narrowing its focus, adapting from a broad, environmental existence into a specialist that lives in and sickens a single host. In other words, ancient DNA caught salmonella in the act of becoming a human disease. What drove that change? The answer points straight at how people had begun to live. The rise of farming packed humans into denser, more permanent settlements. It brought them into constant, intimate contact with herds of domesticated animals. It concentrated human and animal waste near the water and food supply, the perfect conditions for a bacterium that spreads by the fecal-oral route. This was the Neolithic demographic transition, the population boom that farming unleashed, and it built the exact environment a crowd-adapted pathogen needs. Closely related strains turning up across wide distances show the bug then traveled with people as they moved, riding the same step expansions that carried other diseases. The genomes came from people tied to some of prehistory's great movements, including populations associated with the corded ware culture and the steppe pastoralists, whose expansions reshaped the genetics of Europe. That geographic spread matters, because closely related strains recovered from distant sites imply the bacterium was not evolving in one isolated spot, but traveling with human beings as they migrated and traded. Systemic infections like paratyphoid fever seed the bloodstream and, through it, the bone marrow, which is one reason their DNA can survive in skeletons at all, unlike a gut infection that might leave no lasting trace in the bones. By screening large numbers of ancient individuals, the researchers could pinpoint roughly when the human-adapted lineage split away from its broader host ancestors, placing the birth of this particular fever squarely in the age of the first farmers and herders. Mainstream researchers read this as a pathogen crowd adapting to the new farming world. Others emphasize the animals themselves as the source, a zoonosis handed over through herding. Some point to trade routes seeding the strain across the continent. However it happened, the lesson is stark. The first villages did not just give humanity bread and beer. They gave it a fever that lived in the water people drank. And the deepest scars left by these ancient plagues are not in the water or the soil. Some of them are inside you right now. Number 5. The plagues written into Neanderthal DNA. 50,000 years ago, on a cold Eurasian plain, a Neanderthal is dying of a virus, racked and feverish, one of countless deaths in outbreaks we will never name. But that dying population had something our ancestors lacked, and when the two groups met and had children, the Neanderthals handed over a genetic inheritance that still decides, today, whether you live or die when a virus finds you. Every person alive whose ancestry lies outside Africa carries roughly 1.5% to 2% Neanderthal DNA. The legacy of interbreeding around 50 to 60,000 years ago, when modern humans expanding out of Africa met Neanderthals who had already lived in Eurasia for hundreds of thousands of years. For a long time, that DNA looked like a genetic curiosity. Then, in 2018, David Enard and Dmitry Petrov published a study in the journal Cell that changed how we see it. They focused on what they called virus-interacting proteins, the human proteins that physically grapple with viruses during an infection. And they found something remarkable. The Neanderthal DNA surviving in modern Europeans is heavily concentrated in proteins that interact specifically with RNA viruses. More than 150 such proteins bear a Neanderthal signature. This is adaptive introgression, the flow of useful genes from one species into another through hybridization. The logic is chillingly elegant. Neanderthals had spent ages adapting to the pathogens of Eurasia. Modern humans arriving from Africa walked into a landscape full of diseases their immune systems had never faced. Interbreeding was risky, an exchange of unfamiliar germs, but it also handed the newcomers a set of gene variants, already tuned to fight local viruses. Researchers have described it as a poison and antidote at once. Other work ties Neanderthal ancestry to specific immune genes, the OAS antiviral cluster, and the toll-like receptor genes, some of which crank up inflammation, protective against infection, but linked today to allergy and autoimmune disease. The statistical case is strong precisely because the pattern repeats across independent human populations, rather than appearing as a fluke in one group. And Neanderthals were not the only archaic humans to leave us such gifts. The Denisovans, a sister population known mostly from a finger bone and a few teeth, contributed immune-related variants of their own to the ancestors of people in Asia and Oceania. The emerging picture reframes interbreeding entirely. It was not just a mixing of ancestry, a blending of faces and builds. It was a transfer of hard-won immunological experience, tens of thousands of years of adaptation to Eurasian disease, handed to newcomers who would otherwise have faced those pathogens blind. The price was exposure to whatever the archaic humans carried. The reward was a head start against an entire continent's worth of germs. This is not ancient trivia. During the recent coronavirus pandemic, one Neanderthal inherited stretch of DNA on chromosome 3 was linked to a higher risk of severe illness, while another, in the OAS region on chromosome 12, offered protection. The epidemics that helped kill the Neanderthals are still shaping who survives a modern outbreak. Because RNA viruses leave no fossils, these scars in our genome are among the only records that those ancient plagues ever happened at all. And sometimes, the record is not in our genes, but in a single cave full of the dead. Number 6. Aveline's hole. At the mouth of a limestone cave in a green English gorge, in the cool light spilling through the entrance, the floor is lined with the bones of dozens of people, laid to rest in the sediment more than 10,000 years ago, the largest gathering of the dead from that age anywhere in Britain, and no one is certain why they are all there. The cave is Aveline's Hole in Burrington Combe in the Mendip Hills of Somerset. It is the earliest scientifically dated cemetery in the whole of Britain. Radiocarbon dates place the burials at around 10,200 to 10,400 years ago, in the early Mesolithic, the era of hunter-gatherers who lived after the last ice age released its grip. Estimates of how many people were buried there range from 21 clearly documented to perhaps 50 or more originally. The site was found in 1797, when two men digging into the hillside broke through and discovered a chamber full of human bone. Tragically, much of the original collection was stored in Bristol and destroyed in 1940, when wartime bombing hit the building holding it. What survives are some bones and the careful records made before the loss, including early antiquarian notes. Those records describe grave goods, perforated animal teeth, and fossil ammonites, and the red ochre associated with the dead. Isotope analysis of surviving bone suggests these people ate a land-based diet, despite living within reach of the sea. Modern study of the site has been carried on largely by the University of Bristol Speleological Society, together with academic collaborators, working from the surviving fragments and the historical documentation. Their radiocarbon dates are what pin the burials so firmly to the early Mesolithic, making this not just an old cemetery, but the oldest one known in Britain by a wide margin. The grave goods hint at care and ceremony. The perforated teeth perhaps worn as ornaments, the fossil ammonites gathered deliberately, the red ochre scattered over the dead in a practice seen across Ice Age and post-Ice Age Europe. This was not a careless disposal of bodies. It was a place returned to, or used intensively, by people who marked their dead with meaning, which only sharpens the question of why so many of them came to rest here in so short a window of time. What makes Aveline's Hole so puzzling is the sheer concentration. Mesolithic Britain was home to small, mobile bands of hunter-gatherers, and formal cemeteries from this period are extremely rare. So why are so many people gathered in one cave, in what may be a relatively short span of time? Here the interpretations pull apart. Perhaps it was a dedicated burial place, used steadily over generations. Perhaps it was a gathering site, where scattered bands returned to enter their dead together. Or perhaps something killed a lot of people in a short time. A hard season, a famine, or a disease that swept a community. The truth is that the surviving evidence cannot settle it. These were some of the first people to live in Britain after the Ice Age, in a warming, reforesting world, at a time when Britain was still joined to the continent by the drowned land we call Doggerland. Dozens of them ended up together in the dark, and the reason went into the ground with them. Mass death that leaves no clear cause is the signature of exactly the kind of outbreak that never gets written down. And some of those outbreaks were vast, sweeping across whole regions, tens of thousands of years before the first city. Number 7. The 20,000-year-old coronavirus epidemic. Across a frozen valley in ancient East Asia, a band of fur-clad hunter-gatherers moves through the cold, and something is moving with them that they cannot see. A virus from a family the world will not name for another 20,000 years, when it returns and shuts down the entire planet. The virus is a coronavirus, and the outbreak it caused so long ago is written into the genes of their descendants today. The discovery came in 2021 in a study in the journal Current Biology led by Yasin Sowilmi, Ray Tobler, and Kirill Alexandrov. They took roughly 2,500 human genomes from the Global 1000 Genomes Project and searched for the fingerprints of ancient natural selection. They looked specifically at the genes that build virus-interacting proteins, and among them at proteins known to interact with coronaviruses. In 5 East Asian populations, and largely nowhere else, they found a powerful signal of adaptation. The pattern implies that an ancient coronavirus, or something very like it, swept through the ancestors of East Asians and pressed hard on their survival. Using the molecular clock, the pace at which mutations accumulate, they dated the onset of this selection to roughly 20 to 25,000 years ago. About 42 genes carried the marks of this adaptation, and the signal built up over many generations, suggesting an epidemic that returned again and again, or ground on for a very long time. Some of the very genes flagged are involved in how coronaviruses hijack our cells, and remarkably, several are targets of modern drugs now being explored against the coronavirus behind the recent pandemic. Detecting an epidemic this way has a name, a kind of genomic paleoepidemiology, and it works by spotting the statistical footprint that natural selection leaves when a pathogen kills enough people to change which gene variants survive. The signal here was not a single sharp event, but a sustained pressure, playing out across many generations, which is why researchers suspect the outbreak either recurred in waves or persisted for a very long time. Its concentration in East Asian populations and its near absence elsewhere points to that region as the epicenter, which fits neatly with a modern fact that the bat populations of East Asia remain one of the planet's great reservoirs of coronaviruses. The virus that caused this ancient epidemic is gone, unrecoverable, an RNA pathogen that rotted away tens of thousands of years ago. What remains is its shadow, cast forward through time onto the DNA of everyone descended from those who lived. This is a profound point. Coronaviruses have been hunting humans for tens of thousands of years, long before farming, long before cities, long before writing. This was a truly prehistoric pandemic, and it cannot be read from any virus, because coronaviruses are RNA viruses that leave no trace in our genome directly. It can only be read from the scar the epidemic carved into the survivors, from the shape of the descendants it left behind, the timing around the coldest depths of the last ice age, and the location in East Asia, a long-standing reservoir of bat coronaviruses, all fit. 20,000 years ago, the same viral family that would one day close borders and empty cities was already rewriting the human genome. And prehistoric outbreaks did not just cross generations. Sometimes they crossed entire oceans, riding on the bodies of animals. Number 8. The Seal-Borne tuberculosis of the Americas. On a sunlit stretch of the ancient Peruvian coast, a hunter drags a sea lion up onto dark volcanic rock, and in the animal's lungs, and now on the breeze between them, is a killer that will settle into the hunter's own chest. A disease that was never supposed to reach this continent until European ships arrived. And yet, here it is, 1,000 years too early. The disease is tuberculosis, caused by bacteria of the Mycobacterium tuberculosis complex. Archaeologists had long known that some pre-Columbian Andean skeletons showed the ravages of tuberculosis, including the collapsed, angled spine of Pott's disease. The standard assumption was that any real tuberculosis in the Americas must have come with Europeans after 1492. Then, in 2014, a study in the journal Nature, led by Kirsten Boss and Johannes Kraus, tested that assumption directly by pulling ancient DNA from 3 tuberculosis-scarred skeletons from the Peruvian coast near the Chiribaya, dated to around 1,000 years ago. The genomes they recovered delivered a shock. The closest living relative of this ancient Peruvian tuberculosis was not a human strain at all. It was Mycobacterium pinnipedi, the form of tuberculosis that infects seals and sea lions. The people of this coast had caught their tuberculosis from marine mammals. The proposed story is astonishing in its reach. Tuberculosis infected seals in Africa. Those seals carried it across the ocean as they ranged the southern seas, and when they hauled out on South American shores, coastal peoples who hunted and butchered them caught the disease and passed it among themselves. This overturned the old certainty that European contact introduced tuberculosis to the New World. It also forced a rethink of the disease itself. The molecular clock in the study suggested the entire tuberculosis complex was far younger than anyone had believed, on the order of 6,000 years old, which reshaped debates about how the disease spread with human populations. That young age is genuinely contested, and some researchers argue the clock still underestimates tuberculosis's true antiquity. But the core finding held. After Europeans did arrive, their human-adapted strains largely replaced the seal-derived ones, which is why the marine origin stayed hidden until ancient DNA revealed it. The 3 genomes are among the oldest tuberculosis genomes ever reconstructed from the Americas, and they survived because the disease left lesions in dense bone, and because the cool, arid coastal environment of southern Peru slowed the decay of the DNA inside them. The Chiribaya were a coastal people deeply tied to the sea, harvesting its fish and its mammals, and that intimacy with marine life is exactly what would have exposed them to a seal-borne infection. It is a textbook case of a zoonosis, a disease jumping from an animal reservoir into humans, and a vivid demonstration of how ancient DNA can uncover transmission routes that no one would ever have guessed from bones alone. The finding did not just add a footnote to the history of one disease. It forced scientists to redraw the timeline of tuberculosis across the entire globe. A disease that beat Columbus to a continent by 1,000 years, carried in on the tide and the bodies of seals, is a reminder that prehistoric people faced epidemics welling up out of the animals and the sea around them. And some of the largest die-offs of all may have struck creatures far bigger than any human. Number 9. The Hyperdisease Hypothesis. On a bleak, snow-scoured steppe, a woolly mammoth sinks to its knees and does not rise, its eyes dimming, and out across the white distance, the shapes of other fallen giants lie half-buried. Mammoths and mastodons and ground sloths, a world of enormous animals collapsing together. And one controversial idea says the thing that felled them was not the cold and not the spear, but a plague. This is the hyperdisease hypothesis, argued most prominently by the paleontologist Ross McPhee and the virologist Preston Marks in the late 1990s. It steps into one of the great mysteries of prehistory, the extinction of the great Ice Age megafauna. The animals over about 44 kilograms that vanished across the Americas, Australia, and northern Eurasia toward the end of the last Ice Age. In the Americas alone, more than 35 genera of large mammals disappeared roughly 13 to 11,000 years ago, around the time humans arrived. Mammoths, mastodons, giant ground sloths, armored glyptodonts, native horses, and camels all vanished. The two leading explanations have long been climate change, as the Ice Age world transformed, and overkill, the idea that human hunters wiped them out. The Hyperdisease Hypothesis offers a third possibility. What if humans, or the animals traveling with them, introduced a pathogen so lethal and so broad in its targets that it tore through populations of animals with no prior exposure and no defenses? It would explain something the other models struggle with: a rapid, near-simultaneous collapse of many different species. Here is the pivot that keeps the debate alive. For a single disease to kill dozens of distantly related giants is biologically strange, because most pathogens are fairly specific to their hosts. That is the strongest objection. And direct evidence is desperately hard to find, because recovering the DNA of an ancient pathogen from extinct megafauna is far harder than recovering the host's own DNA. Yet the correlation between the arrival of humans and the pulses of extinction on continent after continent is exactly what a spreading disease would produce. And modern history shows disease can devastate wildlife on a massive scale, from rinderpest scything through African herds to a fungal plague driving amphibians to extinction today. The debate does not sit in isolation either. It runs alongside the overkill hypothesis, championed by the scientist Paul Martin, which credits human hunters with the slaughter, and alongside climate and vegetation models that tie the extinctions to the wrenching changes at the end of the Ice Age. In North America, the picture is tangled further by the younger Dryas, a sudden return to cold conditions, and by contested claims of a cosmic impact around the same time. Disentangling these forces is enormously difficult because they all strike in roughly the same window, and a die-off from one can look much like a die-off from another. What may eventually break the deadlock is the same technology transforming the rest of this story: advances in ancient DNA and in ancient proteins that could one day let researchers screen the remains of extinct giants directly for the pathogens that might have felled them. Most researchers now favor a combination of climate change and human pressure, with disease as a possible contributor rather than the sole cause. But the hypothesis endures because it captures something true and unsettling. Invisible pathogens can reshape whole ecosystems. And if it is right, even in part, then the deadliest outbreak in the entire human story is one that emptied continents of their largest animals, before anyone could record a word of it. All of these outbreaks, human and animal, trace back to a single turning point, the moment our species changed how it lived. Number 10. The first epidemiological transition. The first farmer wakes in the first crowded village, surrounded by more people than a hunter-gatherer might meet in a year, with animals penned close by and grain stored against the wall. And in that ordinary morning is the invisible birth of the epidemic itself, the moment humanity built the world its worst diseases needed in order to exist. Scientists call it the first epidemiological transition. The broader idea of epidemiological transitions was framed by Abdel Omran in 1971, and anthropologists such as George Armelagos applied the concept to the deepest shift of all, the Neolithic Revolution. Before farming, humans lived in small, mobile bands, scattered thinly across the land. That way of life is a poor home for many infectious diseases. The acute crowd diseases, the ones that burn through a population and then need fresh victims, cannot persist in tiny groups because they run out of people to infect. They require dense, settled populations, and farming, beginning around 12 to 10,000 years ago, created exactly that. The Neolithic package assembled every ingredient a pathogen could want. Domesticating animals brought people into daily contact with the diseases those animals carried, and many of our worst infections crossed over from them. Measles appears to descend from rinderpest, a cattle disease. Smallpox, traces to an animal pox virus. Influenza bruise in birds and pigs. Settling in one place, concentrated human waste, fouling water and food and spreading typhoid and dysentery. Stored grain drew rats, and rats brought their own plagues. Irrigation left standing water where mosquitoes bred malaria, and snails carried the parasites of schistosomiasis. The bones of early farmers testify to the cost. Compared with the foragers before them, many Neolithic people were shorter, with more dental disease, and with skeletal marks of anemia and childhood stress, the porous bone of parodic hyperostosis, and the growth lines of enamel hypoplasia. Rising density created unbroken chains of transmission, and trade between settlements linked separate pools of disease into networks an epidemic could race across. There is a grim logic to why the deadliest human pathogens are, in evolutionary terms, relatively young. A disease, like measles, simply could not have existed among Ice Age foragers, because it needs a constant supply of fresh, susceptible hosts, and small mobile bands cannot provide them. Only once populations swelled past a critical threshold could such infections take hold and stay. The same farming revolution that fed the swelling numbers also gave those numbers the standing water, the stored grain, the penned animals, and the accumulated waste that pathogens exploit. Later, in the industrial age, a second epidemiological transition would shift the burden again, away from infection and toward the chronic diseases of long life. But the first transition is the hinge on which the entire human experience of epidemic disease turns. The moment scattered survival gave way to crowded vulnerability. Farming traded one set of problems for another, more food and more children, but a far heavier burden of infection. Mainstream science reads this as populations finally crossing the threshold that crowd diseases require. Paleopathology, the study of disease in ancient bones, provides the hard evidence in the skeletons themselves. And a darker framing asks whether farming was, in a sense, a disease trap, a bargain that fed more people while making them sicker. Whatever the verdict, the conclusion is clear. Every pandemic that history would later record was made possible here by the choice to settle down. The stage for all of it was built in prehistory. Step back from these 10 stories, and a single strange truth comes into focus. We are reading outbreaks that left no writing, and the archives they left instead are astonishing. A woman's germs survived in a lump of chewed tar. A blood virus and a bloodstream bacterium waited in the marrow and the tooth pulp of the buried dead. A tuberculosis strain confessed its marine origin from 3 scarred skeletons. A coronavirus epidemic and a Neanderthal inheritance of defenses were both invisible until we learned to read them not in any pathogen, but in the shape of the survivors, in the human genome itself. And where no genome survives, the archaeology speaks in its own blunt way, in the packed floor of a Provence tomb and the crowded dark of a Somerset cave, telling us that many people died together, and quickly, for reasons the bones alone will not give up. It is worth noting how differently these two kinds of evidence behave. The genome is a record that travels forward in time inside the living, carried in the bodies of descendants who may sit thousands of miles and hundreds of generations from the original outbreak. The archaeology is fixed in place. A single floor, a single cave, a snapshot of one community's worst day. Read together, the moving record and the fixed one. Let researchers triangulate, using the genetic scar to suggest what killed people, and the buried bones to show where and when it happened. Neither alone would be enough. It is the combination that has turned prehistory from a silent age into one that, increasingly, we can hear. The tools that opened these archives, paleogenomics and paleopathology, are barely a few decades old, and they have already rewritten how deep the human disease story runs. And yet, for all we have recovered, the honest measure of this subject is how much remains unknown. We still cannot say for certain whether the dead of Roaix were killed by arrows, or by an epidemic, or by both. We still do not know what gathered so many bodies into Aveline's hole. We may never be able to test the hyperdisease hypothesis directly, because the pathogens that would prove it, if they existed, may have vanished with the giants they killed. And beyond these named cases lies a far larger silence. The unknown number of prehistoric epidemics that swept through human bands and left no trace at all, no tomb, no scarred bone, no lasting mark on the genome, outbreaks that erased people so completely that even their erasure is invisible to us. That silence is not a gap to be shrugged off. It is a reminder that the record we can read is skewed toward the outbreaks that spared enough survivors to leave descendants, or struck communities settled enough to leave graves. The most lethal events of all, by their very nature, are the ones least likely to have left us anything to find. History, in that sense, is written by the epidemics that failed to finish the job. Every new technique, every improvement in extracting DNA from a scrap of tooth or a smear of ancient soil pushes a little further into that darkness, recovering names and dates and pathogens that seemed lost for good. The story of the first outbreaks is still being written, and most of its pages are still blank. What thawing permafrost may yet surrender, and what sharper ancient DNA methods may still coax from a fragment of tooth, could add names to that silence in the years ahead. The deadliest outbreaks in our history may not be the plagues we remember. They may be the ones no one survived to record. If the deep past still has questions for you, there's another investigation waiting on your screen right now, and subscribing keeps them coming.